So we had just finished up the Krebs cycle yesterday. So we saw that the cell makes just a little bit of ATP, right? 2 molecules in glycolysis and 2 molecules in Krebs for a total of 4 net ATP molecules. And they are the result of substrate level phosphorylation, where a phosphorylated compound, an organic compound, will donate its phosphate to ADP to make ATP with the help of an enzyme. So that's one way the cell can make ATP, but the cell makes very little ATP that way. We said that approximately 90% of the ATP made in a cell is the result of oxidative phosphorylation. So today we're going to talk about the electron transport chain in chemiosmosis and see just how all that ATP is made. So we know the cell has stored a lot of potential energy with the electrons taken from glucose and stored in NADH and FADH2. And we said the cell has made 10 NADHs and 2 FADH2 molecules for a total of 12 electron carriers. And they're going to carry those electrons to the electron transport chain to make ATP. And the process here is called oxidative phosphorylation because a lot of redox reactions occur where the electrons are passed from one complex to the next. So one complex becomes oxidized when it transfers electrons to the next complex, which becomes reduced, and so on and so forth. Oxidation, reduction, oxidation, reduction. In the process, we're going to see that hydrogens will get pumped across the membrane and accumulate in a high concentration. And what do we know about molecules in a high concentration? Where do they want to go? Where do molecules in a high concentration want to go? Right, to an area of low concentration. And as we'll see, that movement of hydrogens from high concentration to low concentration is an alternate form of energy. So the electrons, as they're passed from complex to complex, that's a form of energy used to pump hydrogens. And the movement of the hydrogens from high concentration to low is another alternate form of energy. And we'll see how that's used to make ATP. So electron transport chain is in the cristae, right, the inner mitochondrial membrane that's highly folded. And that's the reason why we have all that membrane folded inside of the mitochondria, because that's where the electron transport chains are. They must be embedded in a membrane. And if you fold the membrane, you can pack a lot of transport chains in a small volume. So the transport chain is made up of proteins in multi-protein complexes, and the carriers alternate by being reduced and oxidized as they accept and donate the electrons. So, as I said before, oxidation, reduction, oxidation, reduction. Electrons drop in free energy as they go down the chain. So remember, all that energy has to be released in small amounts, right? So as not to burn up the cell, because heat is being produced and being released. Ultimately, those electrons end up combining with oxygen and some hydrogen to make water. So here we see an example of an electron transport chain. So we have our complexes. You don't need to know what they are. And NADH and FADH2 donate their electrons, but notice they donate them at 2 different locations in the electron transport chain. And that will result in different amounts of ATP being produced, as we will see. But once the electrons reach the end of the transport chain, they have to go somewhere, right? They can't just float around in inner space. And so those electrons combine with oxygen and hydrogen to make water. So this is where the water gets made. Remember the equation, right? The simplified equation. C6H12O6 plus 6O2 yields 6 CO2 plus 6 water, and here's where the water gets made, plus that energy, ATP and heat. So water is a metabolic byproduct. But the whole purpose of oxygen, the whole reason why you and many other organisms need to breathe is that oxygen must be there at the end of the electron transport chain. That's why you need to breathe. That's it. You must have oxygen to pick up those electrons when they reach the end of the transport chain. Otherwise, all of the complexes will get full of electrons and they stop pumping hydrogens. So no more ATP gets made and you die. Your brain dies first, but you'll die. So the whole reason why, again, you need to breathe, you need that oxygen to be the final electron acceptor at the end of the transport chain. That way the electrons continue to flow. Hydrogens continue to be pumped, and the cell can continue to make ATP. So now you're learning why you need to breathe. All that oxygen you take in, it's to serve as the final electron acceptor at the end of the electron transport chain. Now, when it comes to electron transport chains, for eukaryotes, they're all basically the same. When it comes to prokaryotes, some of the fine details differ, like the different cytochromes we see here, cytochrome B, C, A. They are not exactly the same in prokaryotes, and it actually differs from species to species in prokaryotes. So the fine details of electron transport are a little bit different in prokaryotes, but the overall idea and how it works is the same. It's just that the eukaryotic transport chain is consistent from eukaryote to eukaryote. It is not consistent from prokaryote to prokaryote. There are some differences. But the overall process works the exact same way. So when the electrons are donated by the NADH and FADH2, right, they're donated to those complexes in the membrane. As the electrons are passed from complex to complex, hydrogens will get pumped across the membrane. And as we'll see, those hydrogens are going to accumulate in the intermembrane space, the space between the inner and the outer membranes. And because they're accumulating in a high concentration, those hydrogens want to diffuse back through the membrane to an area of low concentration. But the hydrogen ions are positively charged, which means they can't get through the membrane, right? That hydrophobic interior of the membrane prevents the hydrogens from getting through. So you have all these hydrogens in a high concentration, and they want to get through to the other side of the membrane where the concentration is low, but they cannot simply diffuse through the membrane. So let's see how they get through. So the whole purpose of collecting all those electrons, right, the whole purpose of making all the NADH and FADH2 is so the NADH and FADH2 can donate those electrons to the electron transport chain. As the electrons are passed down the chain from complex to complex, that causes hydrogens to get pumped across the membrane into the intermembrane space where they accumulate in a high concentration. So remember, we said that in order to concentrate a substance against its gradient, you need an input of energy to do that. And for most things, that energy is ATP. You can't use ATP here, right? The cell is trying to make ATP, So it cannot use ATP to make more ATP here. Otherwise, there'd be no net gain. So there needed to be alternate forms of energy. One of those is passing electrons from complex to complex. Each time the electrons are passed from complex to complex, a little bit of energy gets released. And that energy is harnessed and used to pump hydrogens against their gradient. And that's why we get an accumulation of hydrogens. So now we have all these hydrogens, a high concentration. We know that they want to diffuse to an area of low concentration. In this case, that's back on the other side of the membrane where they came from. But as I said, the hydrogens cannot simply diffuse through the lipid membrane. So we know that there are, transport proteins, which can help molecules to simply move through the protein to the other side of the membrane. In this case, it's a very special protein. It's an enzyme, ATP synthase. So this is the enzyme that makes ATP. So enzymes need to use some kind of energy in order to do things, right? Normally that energy is ATP. In this case, it cannot be ATP, right? A cell cannot use ATP to make more ATP here, right? Because there would be no net gain. So there needs to be an alternate form of energy. In this case, it's the movement of those hydrogen ions through the ATP synthase. That physical movement of hydrogens is an alternate form of energy. And so ATP synthase uses the movement of hydrogens to catalyze the reaction to make ATP. So think about this. The cell needs to make a lot of ATP. It cannot use ATP to make ATP here. At the very, very beginning of glycolysis, a little bit of ATP must be invested in order to make more. But that was only 2 molecules of ATP. The cell needs to make a lot of ATP. So it can no longer use ATP to make ATP. It's got to use something else. It needs an alternate form of energy. So we have electrons and hydrogens. The electrons are used to pump the hydrogens against their concentration gradient, so the hydrogens accumulate in a high concentration. Then those hydrogens will diffuse through the ATP synthase back to the other side of the membrane where the concentration is low. So we're using a concentration gradient of hydrogens as an alternate form of energy. As those hydrogens move through the ATP synthase enzyme, that's a form of energy that ATP synthase uses to make ATP. So we call this process chemiosmosis because it's a form of diffusion, in this case using hydrogen ions. So that's the chemiosmosis part. But that's how cells make a lot of ATP. So here we see ATP synthase. It looks kind of like a motor, with rotors in it. It's just meant to illustrate the concept here. But you have this high concentration of hydrogen ions in the intermembrane space, which is between the inner membrane and the outer membrane, which is not shown here. Because these hydrogens are in a high concentration, they want to go back to the other side of the membrane where the concentration is low, but again, they can't diffuse through the hydrophobic interior of the membrane. So a special transport protein will allow the hydrogens to diffuse. And that special transport protein is an enzyme, ATP synthase. As the hydrogens move through the ATP synthase, that movement is a form of energy, which allows ATP synthase to form ATP. So questions or comments? So another analogy would be like water, flowing through the turbines of a dam. As the turbines rotate, they create electricity. So our hydrogens are the water. Here we have the turbines. As the hydrogens move through the turbines, it causes them to rotate. Just use your imagination on that one. And that allows this enzyme to make ATP. So questions, comments, all sound good? Where are we in the cell? We're in the mitochondrion, right? So the inner mitochondrial membrane is where the electron transport chains are and where the ATP synthase enzyme is. And so the space between the inner and the outer membranes is the intermembrane space. That's where the hydrogen ions accumulate in a high concentration. And they flow back through the ATP synthase into the matrix, right? Sort of like the center part of the mitochondrion. So a fancy word for the hydrogen ion gradient is the proton motive force. It's named that way to emphasize its capacity to do work. So the proton motive force is the hydrogen ion gradient, a high concentration of hydrogens ions diffusing through the ATP synthase to the other side of the membrane. You're just following your concentration gradient. So it's an alternate form of energy used by cells. So this was some incredible research to determine how all of this worked. Right? The idea that electrons are harvested from food molecules like glucose, and those electrons are used to pump hydrogens, and the hydrogens diffuse through an enzyme, ATP synthase, that catalyzes the formation of ATP. I mean, it's a pretty awesome system when you think about it. It's really awesome to think about how something like this could have evolved, because when we start learning about photosynthesis, it's the exact same idea with these electron transport chains, except the electrons are coming from water in photosynthesis, but they're used, again, to pump hydrogens across the membrane where they will accumulate and flow back through ATP synthase. So photosynthesis uses the same process of electron transport and chemiosmosis to make ATP. Prokaryotes, many bacteria, use this exact same process, only they don't have mitochondria. So where is all this happening? Right, there's no inner and outer membrane because there's no mitochondria. So how can this possibly work in bacteria? Right, the cell membrane, the plasma membrane, that's the only membrane they have, right? So then where do the hydrogens get pumped, to the inside or the outside of the cell in order to accumulate in a high concentration? We have a prokaryotic cell. It just has the cell membrane. So where do the hydrogens go? Are they pumped to the inside or to the outside where they can accumulate in a high concentration? Okay, Chris says to the outside. Does everybody agree with Chris? A 50-50. So H plus Where have you seen H plus before? We've talked about H plus before. H plus, when you see H plus, what's something you should be thinking about? 2 letters. Well, not that letter. That's the OH minus. And what goes along with that? Right, pH. So if you have a high concentration of H plus, what does that mean? Right, acidic. So if the hydrogen ions were pumped into a bacterial cell, that would make the cytoplasm very acidic. So that's probably not a good thing, right? So Chris was right. The hydrogens are pumped to the outside of the cell. And so when you guys go on to take microbiology, you'll learn that there are 2 major cell types, gram-positive and gram-negative cells. And the gram-positive cells have a thick layer of what's called peptidoglycan on top of the cell. And so there's a space in here where the hydrogens can accumulate. In a gram-negative cell, they actually have 2 membranes. And so there's a space in between the 2 membranes. And that's where the hydrogens can accumulate. So the hydrogens do accumulate just on the outside of the plasma membrane in the gram-positive cells, in between the membrane and the thick layer of peptidoglycan. And in the gram-negative cells, they actually have 2 membranes, a double membrane system. So in the space in between the inner and outer membrane, which they call the periplasm rather than the intermembrane space, but the periplasm is where the hydrogens can accumulate. And then they flow back into the cell through ATP synthase again in order to make ATP. And the electron transport chains in bacteria are embedded in the cell membrane. So in the plasma membrane, that's where the transport chains are, right? Because they only have that one membrane, so that's where the transport chains have to be. So here we see the intermitochondrial membrane with all the protein complexes embedded in the membrane. NADH donates to the first complex in the chain, and this complex gets excited. It transports the hydrogens. As the electrons are passed from complex to complex, it causes more hydrogens to get pumped. And when the electrons reach the end of the transport chain, they combine with oxygen and hydrogen to make water. So that's a very important concept. That's why this is called aerobic cell respiration. It's aerobic because oxygen is the final electron acceptor at the end of the chain. That oxygen must be there. If there is no oxygen, electron transport stops. All the hydrogen pumping stops. ATP synthase has no energy source to make ATP. So none of this functions. Electron transport and chemiosmosis does not function under anaerobic conditions. Most of the Krebs cycle does not function. So that leaves the cell with just glycolysis. And remember, only 2 molecules of ATP get made in glycolysis. If you're a single-celled microorganism, that's enough. You can survive. But if you're a complex multicellular organism like a human, you die, right? We can only go 4.5 to 5 minutes depending on our body temperature. If you're super cold, like you drowned under a frozen lake. You can actually go longer than 5 minutes and still be resuscitated without brain damage if you're super cold. But if it's normal, room temperature, you got about 4.5 to 5 minutes before you suffer brain damage and then death without oxygen. So because we need that oxygen to keep the electrons flowing, right? So the cell can make a lot of ATP. And your brain cells especially require a lot of ATP to function. I know it doesn't seem like it when you look at some people, but our brains are very greedy organs. They need a lot of glucose so they can make a lot of ATP to function. So all the hydrogens get pumped, they accumulate in a high concentration, and they follow their concentration gradient from high to low by moving through the ATP synthase enzyme. As the hydrogens flow through the ATP synthase, that movement of hydrogens is an alternate form of energy that ATP synthase uses to make ATP. So this whole process is the oxidative phosphorylation to make ATP. Questions, comments? Okay, I have a couple of videos to show you, so I'll turn down my volume a little bit. And so these were working in the Blackboard page this morning, so I'm going to go there to show you rather than going to YouTube. That way we can avoid all the advertisements and commercial interruptions. So these are in the Blackboard page.
As this mountain biker heads up the trail, the breakfast he ate this morning is being burned to power his bike ride. His breathing rate increases as his leg muscles demand more oxygen to burn more fuel. Let's zoom down to where this fuel is burned, our cells. Here, the blood vessel on the left delivers fuel and oxygen to a single muscle cell. In cellular respiration, energy and fuel is converted to ATP, shown here as starbursts. Most ATP is made in the cell's mitochondria. ATP powers the work of the cell, such as contraction. Let's take a closer look at how ATP is produced from a molecule of glucose, our fuel. Only the carbon skeleton is shown to keep things simple. The first step is called glycolysis, and it takes place outside the mitochondria. To begin the process, some energy has to be invested. Next, the molecule is split in half. Now, the molecule NAD+, an electron carrier, picks up electrons and hydrogen atoms from the carbon molecule, becoming NADH. Keep track of the electron carriers. They play an important role by transporting electrons to reactions in the mitochondria. In the final steps of glycolysis, some ATP is produced, but not much. For every glucose molecule, only 2 net ATPs are produced outside the mitochondrion. However, glycolysis has produced pyruvic acid, which still has a lot of energy available.
I don't know why I got stuck. Oops.
Acid molecule into a mitochondrion has a lot of energy available.
It's getting hung up at this spot for some reason. See, I hate when this stuff doesn't work. All right, we'll have to go to YouTube then and watch commercials.
Grammarly does more.
Jeez.
The breakfast he ate this morning is being burned to power his bike ride. His breathing rate increases as his leg muscles demand more oxygen to burn more fuel. Let's zoom down to where this fuel is burned, our cells. Here, the blood vessel on the left delivers fuel and oxygen to a single muscle cell. In cellular respiration, energy and fuel is converted to ATP, shown here as starbursts. Most ATP is made in the cell's mitochondria. ATP powers the work of the cell, such as contraction. Let's take a closer look at how ATP is produced from a molecule of glucose, our fuel. Only the carbon skeleton is shown to keep things simple. The first step is called glycolysis, and it takes place outside the mitochondria. To begin the process, some energy has to be invested. Next, the molecule is split in half. Now, the molecule NAD+, an electron carrier, picks up electrons and hydrogen atoms from the carbon molecule, becoming NADH. Keep track of the electron carriers. They play an important role by transporting electrons to reactions in the mitochondria. In the final steps of glycolysis, some ATP is produced, but not much. For every glucose molecule, only 2 net ATPs are produced outside the mitochondrion. However, glycolysis has produced pyruvic acid, which still has a lot of energy available. Let's follow this pyruvic acid molecule into a mitochondrion to see where most of the energy is extracted. As the molecule enters the mitochondrion, 1 carbon is removed, forming carbon dioxide as a byproduct. Electrons are stripped, forming NADH. Coenzyme A attaches to the 2-carbon fragment, forming acetyl-CoA. Coenzyme A is removed, and the remaining 2-carbon skeleton is attached to an existing 4-carbon molecule that serves as the starting point for the citric acid cycle. The new 6-carbon chain is partially broken down, releasing carbon dioxide. Several electrons are captured by electron carriers, and more carbon dioxide is released. The carbon dioxide that you exhale comes from the reactions of cellular respiration. 2 ATPs are produced by the citric acid cycle for each molecule of glucose. At this point, only a small number of ATPs have been produced. However, more energy is available in the electrons that are being transported by electron carriers. While the citric acid cycle starts another round, let's follow an electron carrier to the next step in the process. Electron carriers such as NADH deliver their electrons to an electron transport chain embedded in the inner membrane of the mitochondria. The chain consists of a series of electron carriers, most of which are proteins that exist in large complexes. Electrons are transferred from one electron carrier to the next in the electron transport chain. Let's take a closer look at the path electrons take through the chain. As electrons move along each step of the chain, they give up a bit of energy. The oxygen you breathe pulls electrons from the transport chain, and water is formed as a byproduct. The energy released by electrons is used to pump hydrogen ions, the blue balls, across the inner membrane of the mitochondrion, creating an area of high hydrogen ion concentration. Hydrogen ions flow back across the membrane through a turbine. Much like water through a dam, the flow of hydrogen ions spins the turbine, which activates the production of ATP. These spinning turbines in your cells produce most of the ATP that is generated from the food you eat. The process you've just observed, cellular respiration, generates 10 million ATPs per second in just one cell. That ATP can power a biker up the trail, or it can power your brain cells as you learn challenging biology topics.
Yes, that comes from the publisher. So I remember that they were showing you one of the pyruvates going through the Krebs cycle. Remember, there's 2 of them, so everything happens twice. But it's a nice animation of the process, but a really outstanding animation is this next one. And it's just the electron transport chain. It is going to give more detail on what you need to worry about, but it's just an excellent animation, for showing you the process.
All eukaryotic cells, from yeast to those that make up the human body, contain membrane-bound organelles with specialized functions. functions. Mitochondria are double-membraned organelles that harness most of the energy that cells need to grow and reproduce. Nearly all of this energy comes from reactions that take place at the inner mitochondrial membrane. One of the key roles of this membrane is to act as a barrier to positively charged particles called protons, thus allowing a concentration gradient to be maintained where the intermembrane space has far more protons than the matrix. The membrane also contains a large protein complex called the F1-F0 ATP synthase, which uses the proton gradient to drive the synthesis of ATP molecules. These ATP molecules ultimately provide the energy for most of the cell's reactions. Just as man-made power plants produce electrical energy by using the flow of wind, water, or steam to rotate a turbine, the synthase makes ATP by using proton flow from one side of the inner membrane to the other to rotate protein subunits. If there is no proton gradient, synthase subunits stop rotating, and the cell can quickly become starved of energy and die. Therefore, the protein complexes and small molecules that establish this gradient and maintain it play an essential role in the life of the cell. At the heart of this system are 4 protein complexes, numbered 1 through 4. Complexes 1, 3, and 4 directly pump protons from the matrix into the intermembrane space. Complex 2 does not directly pump protons, but it does promote proton pumping in complexes 3 and 4. Proton pumping requires energy, and the 4 protein complexes get this energy by transferring electrons through a series of coupled reactions. This linked process of electron transport is why the 4 complexes are collectively referred to as the electron transport chain. Let's focus on complex 1. A byproduct of sugar metabolism called NADH deposits 2 high-energy electrons in complex 1, where they are passed along a chain of redox centers. Redox centers are clusters of atoms that have different affinities for electrons based on their unique atomic configurations. Let's closely consider a pair of redox centers to reveal 2 reasons why an electron moves from the top redox center to the bottom. First, the bottom redox center has higher affinity than the top one. Second, the distance between these adjacent redox centers is ideal for an electron jump to occur, which explains why electrons typically don't bypass the bottom redox center. A small amount of energy is released each time an electron is passed between redox centers. Complex 1 harnesses this energy across all the redox centers and uses it to pump protons. The last redox center in complex 1 donates 2 electrons to a coenzyme Q molecule. Complex 2 is similar to complex 1 in two important ways. First, high-energy electrons also enter complex 2 via a byproduct of sugar metabolism, although here the molecule is FADH2. Second, complex 2 also transfers electrons between several redox centers before donating them to coenzyme Q. One major difference, however, is that complex 2 does not use the energy liberated to pump protons. Coenzyme Q molecules from complexes 1 and 2 donate their electrons to complex 3. One electron is recyclable and can re-enter complex 3 later, but the other passes through two redox centers before reaching cytochrome C. Cytochrome C carries the electron to complex 4. The electron transport chain ends in complex 4, where a series of reactions involving 4 electrons converts a molecule of oxygen to 2 molecules of water. The proton gradient is strengthened because 4 protons from the matrix are incorporated into water molecules, and another 4 are pumped into the intermembrane space. In the absence of oxygen, the electron transfer comes to a halt, meaning that ATP synthesis also stops. Indeed, the reason we breathe oxygen is so that it can serve as the final electron acceptor at the end of the electron transport chain. In this animation, we have explored each protein complex in isolation, but in reality they are very densely packed. Together, they effectively make the entire surface of the inner mitochondrial membrane a giant cellular power plant.
So a beautiful animation, a little more detail than what we need, but still. And then I got to jump back to where we were. I'm just getting back to the slide where we were. Okay, so some really beautiful animations illustrating the main idea that our original glucose molecule is ripped apart carbon by carbon. Electrons are captured by the NADH and FADH2. Those electrons are donated to the electron transport chain. As they're transported from complex to complex, that movement of electrons gives those complexes the energy they need to pump hydrogens across the membrane against their gradients where they accumulate in a high concentration. Those hydrogen ions want to go back to the other side of the membrane where the concentration is lower, but they can't diffuse directly through the membrane, but they can diffuse through the ATP synthase enzyme. And the physical movement of hydrogens is an alternate form of energy, and that allows ATP synthase to make ATP. And the whole reason why it's aerobic cell respiration is because oxygen must be there at the end of the transport chain to pick up those electrons. That allows the electrons to continue to flow. If there's no oxygen, electron transport stops, and eventually the cells will die. And you heard in the first video the incredible numbers of ATPs that are produced every second. So it's just, I mean, an unfathomable number of ATPs produced in your body every single day. It's just incredible, the numbers of ATP. So ADP is quickly regenerated into ATP so the cell can continue to function and use the hydrolysis of ATP to make essential reactions exergonic, so they will happen spontaneously. So questions or comments about the videos? All good stuff, right? So most of the energy flows from glucose to NADH, a little bit goes to FADH2 in the electron transport chain, which creates the proton motive force, the hydrogen ion gradient, which leads to the formation of ATP. And so I said before, only about 40% of the energy in that glucose molecule is captured and used to make ATP. The other approximately 60% is lost as heat. So that heat energy increases the entropy of the universe, but also that heat energy can be used by animals like us, right, to maintain a constant internal body temperature. Now, I'm going to talk about how many ATPs are produced, and the numbers that I'm going to give you are the maximum theoretical yields. So that's if the system of making ATP were perfect, absolutely no waste. No such systems exist. But if everything were perfect, we get a maximum 36 to 38 molecules of ATP. In reality, it's somewhere around 29 to 30 because no system for making energy is perfect. And so there is some waste. So in reality, the amounts I'm going to show you are lower, but it's easier to talk about the theoretical yields because that's what we've been talking about for many, many years. So remember, glycolysis produces a net ATP production of 2 molecules. In the Krebs cycle, we have 2. And then in the electron transport and chemiosmosis, the oxidative phosphorylation part, we've got 32 to 34 ATPs for a grand total of 36 to 38. And that's maximum theoretical yield. Typically, the 36 molecules of ATP are what they talk about for eukaryotes, and prokaryotes about 38 molecules of ATP. And that's because there's no mitochondria in prokaryotes. So in eukaryotes, substances have to be transported into and out of the mitochondrion, and that uses a little bit of energy. And so the ATP yield is a little bit lower in a eukaryotic cell compared to a prokaryotic cell. But in reality, these numbers represent maximum theoretical yields, right? So they're actually lower than this, but we're not going to worry about those numbers. So to summarize things, in glycolysis, again, 2 molecules of ATP produced and 2 molecules of NADH. In the intermediate step, that decarboxylation step where pyruvic acid is converted to acetyl coenzyme A, no more ATP is made, but 2 molecules of NADH are produced. In the Krebs cycle, we got 2 molecules of ATP, 6 molecules of NADH, and 2 molecules of FADH2. So a total of 4 ATPs, 10 NADHs, and 2 FADH2s. All right, so that's what gets made and how much of it gets made. Now, this chart or this table is a little bit confusing for students because all these numbers that we see in this table, these are numbers of ATPs. So these are ATPs in this table. We know we got 2 ATPs from glycolysis and 2 ATPs from the Krebs cycle. Right, so a total of 4 ATPs produced by substrate level phosphorylation. When it comes to oxidative phosphorylation, the NADHs, remember that in glycolysis, 2 molecules of NADH were produced. Those 2 molecules of NADH yield 6 ATPs. In the intermediate step, again, 2 molecules of NADH are produced. They yield 6 ATPs. And in the Krebs cycle, we said that 6 molecules of NADH were produced, and they yield 18 ATPs for a grand total of 30 ATPs from the NADH molecules. Remember, there are 10 NADHs produced. They yield 30 ATPs. So how many ATPs per NADH? Yield, makes. Results in, produces, makes. So, right, so Zoe says 3, right? So for every NADH, 3 molecules of ATP are produced, right? Because we got, right, 10 NADH, and they yield, or they result in 30 ATPs. So for every NADH, you get 3 ATPs. Now for the FADH2, we're going to get 4 molecules of ATP. And remember, there were only 2 molecules of FADH2 produced, and they yield 4 ATPs. So how many ATPs for every FADH2? 2. So the NADH can produce 3 ATPs, and FADH2 produces 2. Why the difference? NADH and FADH2 are both donating electrons to the transport chain. Why do you only get 2 ATPs from FADH2, but 3 ATPs from NADH? They're both donating electrons, right? So why the difference? Right, 3 ATPs from NADH, 2 ATPs from FADH2, but they're both donating electrons to the transport chain, so why don't you get the same number of ATPs? Well, here's your answer, so explain it. Well, you can see. What's the explanation? So somebody give me something. Chris?
I guess the only thing I could think of is NADH is donating more electrons per.
Not more. But notice where they donate. the NADH donates at the very beginning of the electron transport chain. So the maximum number of hydrogens, which we'll just say is 3, each hydrogen produces an ATP. But the FADH2 donates later, which means only 2 hydrogens get pumped, and they produce 2 ATPs. So even though NADH and FADH2 both donate electrons to the transport chain, they donate at different points. And that results in different numbers of hydrogens being pumped. And it's the hydrogens moving through the ATP synthase that gives the ATP synthase the energy it needs to make ATP. So more hydrogens, more ATP. Fewer hydrogens, fewer ATPs. So does that make sense? I mean, that would make an excellent exam question, don't you think? When the electrons are donated at the beginning of the transport chain, the maximum number of hydrogens get pumped, which means the maximum number of ATPs get made. When you donate electrons later in the chain, fewer hydrogens and therefore fewer ATPs. And that's why we see the difference between right, the amount of ATP produced by NADH and FADH2. Now remember, these are maximum theoretical yields. In reality, it's less. So like for NADH, it's more like 2.5 ATPs, and FADH2, it's more like 1.5, but you know, that it's just harder to think about rather than whole numbers. So, and that's just because no system is perfect. There's always some waste, you know, some loss of energy that's not being used. So fun fact, the energy used by human cells requires the hydrolysis of 100 to 150 moles of ATP daily. How big was a mole? How many atoms or molecules in a mole? Yes, 6.02 times 10 to the 23. So 100 to 150 moles. So 150 times 6.02 times 10 to the 23. So it's an incredibly huge number, just an unimaginable number of ATPs produced every single day. And if you were to weigh those ATPs, they would weigh anywhere from 50 to 75 kilograms. Right, that's the average body weight of a person. You know, an average sized person. A human will typically use up his or her body weight of ATP over the course of a day. Each equivalent of ATP is recycled 500 to 750 times during a single day, right? Because the ATP is hydrolyzed to ADP, which is quickly regenerated back into ATP, 500 to 750 times every day for an incredible amount of ATP. It's just unbelievable, the numbers of ATP. But that's what's required for a complex multicellular organism like a human in order to function. And that was made possible by the evolution of mitochondria. So we said this whole process is aerobic. Oxygen is the final electron acceptor at the end of the transport chain to pick up those electrons and keep those electrons flowing. But glycolysis doesn't need any oxygen to function, right? So glycolysis is actually an anaerobic process. No oxygen is required for glycolysis to occur. So one form of anaerobic respiration is fermentation, which we'll talk about. There are other types of anaerobic respiration. We'll just mention that they exist, but we're not going to go into them. In general, anaerobic respiration, it can use an electron transport chain, and it has some other type of electron acceptor, not oxygen because it's anaerobic, so something like sulfates, carbonates, nitrates can act as electron acceptors in anaerobic respiration. But in general, the process is less efficient and you get less ATP. Oxygen yields the maximum amount of ATP. The fermentation is a form of anaerobic respiration. It uses substrate level phosphorylation, so only those two molecules of ATP produced in glycolysis are available as the energy source. So there's no electron transport chain, there's no Krebs cycle, it's just glycolysis. And so we said only two molecules of ATP get produced, and that's not much. But there is some NADH produced, right? Those two molecules of NADH. But because we're just using glycolysis, the NADHs are not going to carry those electrons to an electron transport chain. The NADH and those electrons are going to be used to reduce and produce the end product of fermentation. And there are many different possible end products depending upon the microorganism. But alcohol fermentation and lactic acid fermentation are two common types of fermentation. So in alcohol fermentation, the pyruvate, right, the pyruvic acids, will be reduced in order to produce the alcohol, the ethyl alcohol. In the process, carbon dioxide will be released. That's why your beer is foamy, right? Why your wine or your champagne is bubbly, right? Because of CO2. Yeasts are single-celled eukaryotic microorganisms that ferment in order to produce alcohol and CO2. And we use yeasts, to make beer and wine. We also use it to make bread. And the reason why that dough rises is because the CO2 bubbles get trapped in the dough and cause the dough to rise. And that's what makes your bread lighter and fluffier with little, air pockets inside when you cut into it. That was caused by the CO2 gas that was trapped in the dough. But these are eukaryotic organisms that ferment. Many prokaryotes also ferment to produce a wide variety of things, including lactic acid. In fact, we use bacteria to spoil milk in a good way, right? When you mix different kinds of bacteria with milk, you get buttermilk, you get yogurt, you get sour cream, all the result of microbial action, to ferment milk and spoil it by making lactic acid, which curdles the milk to make it thick. So here we just see the idea where glycolysis is used to make two molecules of ATP and two molecules of NADH, but the NADHs are used to reduce and produce the end product, which is ethyl alcohol. So if you mix yeast and sugar, they will make alcohol. But they can only make about 4% alcohol by volume, and then they poison themselves and die, right? Because the alcohol is toxic. And so natural fermentation cannot produce concentrated forms of alcohol. In order to make your concentrated forms like whiskey, vodka, bourbon, you have to distill the alcohol to concentrate it. So natural fermentation products like beer and wine are limited in terms of how much alcohol can be present because it poisons the yeast cells that are producing it. So if you want to increase the strength of your alcohol, you have to distill it, boil it off and then condense it in order to get more concentrated alcohol. Any questions about alcohol fermentation? I know most of you don't know anything about alcohol, right? Because most of you are too young, right? Most of you. In lactic acid fermentation, the pyruvate is reduced, by the NADH to form lactate, also known as lactic acid. No CO2 gets produced with lactic acid fermentation. And as I mentioned before, the lactic acid is used to make things like cheese and yogurt. So fungi are often used to make cheese. Does anybody like blue cheese salad dressing? Nobody likes blue cheese? Yeah, the chunks of blue are actually mold. So you're eating moldy cheese. Now, our cells, our muscle cells, are actually capable of performing fermentation, and they perform lactic acid fermentation. So if you're working out at the gym, which most of us have not been able to do because of COVID, but if you were working out at the gym and lifting weights, you know that as you lift those weights, you reach a point where your muscles fatigue and you can no longer lift that weight. You have to rest for a couple minutes right before you can start lifting again. And the reason why, as you're lifting those weights, initially you're able to get enough oxygen to the muscle cells to make those 36 to 38 molecules of ATP. But as you keep on working those muscles, you exceed the capacity to get enough oxygen into those cells to make all of the ATP needed for muscular contraction. So at some point, your muscle cells switch to lactic acid fermentation. At that point, they're only making 2 molecules of ATP per glucose. Those 2 molecules of ATP are not sufficient to maintain muscular contraction, right? Not sufficient for that muscle to continue to be worked. And so you have to stop and rest and get enough oxygen back to those muscle cells to start making all the ATP needed for muscular contraction. And so the lactic acid, it is an acid, so if you've ever heard the expression, feel the burn, that's what you're feeling, lactic acid fermentation. Lactic acid is being produced, and it causes a burning sensation. Eventually the lactic acid will be consumed by the cells and it'll go away, but you can't continue to do work without oxygen. But your muscles, they fatigue. You just can't do much work with lactic acid fermentation. So we need lots of oxygen in our bodies and all of our cells, especially brain cells, in order to make at least 36 molecules of ATP per glucose. And that way we can function and survive. So just showing the production of lactic acid. So again, the electrons in the NADH are going to be used to reduce and produce the end product of fermentation. So you still need that glucose, whether you're using glycolysis or aerobic cell respiration. A glycolysis is considered a universal process. Basically, every form of life, or almost every form of life on the earth, can use glucose in glycolysis. But remember, we're talking about maximum theoretical yields of ATP here. 36 to 38 is the maximum theoretical yield. It's really more like 29 or 30 molecules per glucose. But aerobic respiration is the most efficient. You get the most ATP per glucose if you have oxygen and can use it. Anaerobic microorganisms, they rely on fermentation or anaerobic respiration to produce ATP, but not as much as aerobic respiration does. Anaerobic is just not as efficient, even though that was the first type of respiration to evolve. There are microorganisms called obligate anaerobes or strict anaerobes sometimes, and they use fermentation and anaerobic respiration. Not only do they not use oxygen, oxygen kills them. So oxygen is actually one of the most toxic substances on the earth. It's incredibly toxic, and that's because of free radical formation. And when you take microbiology, you'll learn how organisms deal with those toxic oxygen-free radicals. But there are forms of life on the earth that are killed with exposure to the air. They cannot tolerate oxygen. But there are other microorganisms like yeast and many bacteria, which are facultative anaerobes. And so that means that they will use oxygen if it's present, and they love it, and they'll grow best with oxygen. But if there is no oxygen, they're okay. They will switch to fermentation, and they're just fine, right? So they can survive as a single-celled organism on just 2 molecules of ATP per glucose. But if oxygen is there, they will use it and they will grow better. But they don't need it to survive. So facultative anaerobes, they can switch from aerobic respiration to fermentation. So as I mentioned, glycolysis is a universal process. It probably is one of the first forms of respiration to evolve, right? Because there was little or no oxygen in the atmosphere. When photosynthesis evolved and oxygen was produced as a byproduct, it began to accumulate. And the obligate anaerobes had a choice: evolve to use oxygen or die. And so some of them chose to evolve to use oxygen. Others chose to go hide and stay away from the oxygen so they wouldn't be killed. But if they're exposed to any oxygen, they will die. So they must exist in environments that don't have any oxygen. So when it comes to glycolysis and the citric acid cycle, all those intermediate compounds actually serve a purpose in other reactions in the cell, not just glycolysis and Krebs, right? So they do other things and serve as intermediates for other metabolic pathways, but they're very important in terms of making energy. Remember, catabolism breaks down food molecules to produce energy. Anabolism uses that energy to build, to create macromolecules from smaller pieces. But glycolysis not only uses glucose, it can use other carbohydrates as well. And proteins can be broken down into amino acids and then used to make energy. And the same thing with fats. Fats can also be used to make energy. And so remember that fatty acids, or the fats, are made of glycerol and fatty acids. And the glycerol contributes to energy production, and the fatty acids contribute to energy production. And I think we mentioned this before, that we get more than twice as many calories from a gram of fat as we do either proteins or carbohydrates, because proteins and carbohydrates basically just have one subunit, either the amino acid or the monosaccharides. But a fat molecule has 2 components, both glycerol and fatty acids, and each one contributes to energy. And so we use fat to store energy because it's a much better molecule for storing it than either carbs or protein. And we burn fat, for that energy as well. But the weird thing is that when your body needs energy, it doesn't just use fat first. It uses fat, glycogen, and protein simultaneously. So when you're dieting and exercising, you have to be careful to get enough, protein in your diet, because your body will start to break down muscle and use it for energy, even though you still got plenty of fat. So it would be nice if we could just use all the fat first, then use the glycogen stored in our muscles and livers, and then, burn protein as a last resort. But that's not what your body does. It uses all three pretty much simultaneously. And sort of a fun fact, an oxidized gram of fat produces more than twice as much ATP as an oxidized gram of carbohydrate or protein. And that's approximately 130 ATPs, per lipid molecule. And that's why we store energy as fat. It's a much better way to store to store energy. So again, just keep in mind that not only carbohydrates are used to make ATP, but fats and proteins can also be used. So anabolism will build larger molecules from small molecules, and these small molecules may come directly from the food we eat. It may be molecules that are produced as intermediates in glycolysis or the Krebs cycle. Feedback inhibition works with ATP production, just as it does with many other forms of end products. So it is possible, if you've made too much ATP, that ATP can feed back to one of the enzymes in aerobic cell respiration, bind to it and turn it off. Although that doesn't happen very often, right? Because we use so much ATP, but also ATP production can be increased. And so your body does adjust its metabolism as needed, unless you force it to do something it's not supposed to do. Like if you constantly overeat and consume far more calories than you need, you'll store a lot of excess fat. And then your body gets used to having that excess fat and doesn't want to give it up. And it's one of the reasons why dieting can be so difficult, especially if you yo-yo diet, you starve yourself to lose weight, then put it all back on and starve yourself again. It gets harder and harder to lose that weight because your body is basically getting mad at you. It's saying, you gave me all these calories, I stored all this fat, I like being this way, and now you're forcing me to burn that fat. And your body gets more and more conservative, and it gets harder and harder, to lose weight if your diet, if your weight up and down and up and down. But you can increase your metabolism as a way to produce more body heat. So if it's cold and you're cold, your metabolism actually increases as a way to produce more heat. If it's hot and you're hot, your metabolism slows down so that you make less heat when you burn that glucose. And so your body regulates its metabolism constantly. If you happen to eat, a few excess calories during the day, your metabolism will actually increase without you knowing it, so you burn off those excess calories to maintain that constant body weight. If you don't consume quite enough calories in a day, your metabolism will slow down so you don't lose weight. And so your metabolism will adjust within a narrow range. But like I said, if you keep overeating and storing excess fat, your body says, okay, you clearly want me to maintain this higher body weight, so that's what I'm going to do now. And that's why it's so much harder to lose weight, right? The more weight you gain, the harder it is to lose it. And if you lose it and gain it and lose it and gain it, that makes your body angry at you and makes it very hard to lose that weight. So eat sensibly, exercise sensibly, to maintain a body weight or to lose some weight, but don't, go nuts, because it's not going to work. That TV show, what was that, The World's Biggest Loser or something, where they took a bunch of overweight people and trained them insanely and starved them in order to get them to lose weight. That was a terrible show and the absolute worst possible way to lose weight. And if you go look it up, you'll find that almost every single person on that show regained all the weight, because the way they lost it was wrong. That's not how you lose weight. And so That's a really terrible example for people out there watching that show. Yeah, that's not how you lose weight. So again, just showing you phosphofructokinase is one of the main enzymes involved in the regulation of ATP production. So it's an allosteric enzyme. ATP can feed back to phosphofructokinase, bind to it, and change the shape, which changes the shape of the active site, so it no longer functions. And that will temporarily slow down the production of ATP. But you do burn so much ATP, this is probably not going to happen very often. But this is how ATP can actually serve as a mechanism to reduce the production of more ATP by feeding back to an enzyme, which is phosphofructokinase, binding to it and turning it off. As the cell consumes the ATP, it comes off the phosphofructokinase, and the enzyme goes back to its normal shape and starts to function so the cell can make more ATP. But it's just one example of how the formation of ATP can be regulated inside of a cell. So it's 3:25, so let's just go through these next few slides, and then we'll take a 10-minute break and start lab, because we covered most of the lab stuff last time, so we're just going to go over it again and learn how to do graphing and stuff. So mitochondria and chloroplasts, the powerhouses of the cells. We just learned how mitochondria produce incredible amounts of ATP every second of every day. And your cells have many mitochondria in them, right? Because your cells need a lot of ATP to function. And chloroplasts are another way to make ATP using photosynthesis, using light energy. And so the question was, where did mitochondria and chloroplasts come from? And so a theory developed called the endosymbiotic theory, which tries to explain the origin of mitochondria and chloroplasts. And the idea is that one ancient prokaryotic cell was engulfed by another. But instead of eating and digesting the cell that was engulfed, they actually learned to live together symbiotically. And because all of the other needs of the symbiont were being taken care of by the host cell, that symbiont specialized to do one thing: make ATP. And this, of course, took many, many eons of time for this to happen. But that's how we believe eukaryotic cells developed, from prokaryotic cells that engulfed each other. And then the cell that got engulfed specialized for energy production, either becoming a mitochondrion, or if the cell had the ability to perform photosynthesis, it evolved into a chloroplast. And because those cells now had the ability to make huge amounts of ATP, those cells were able to associate together and start to form animals. And so without mitochondria and chloroplasts, multicellularity would not be possible. Without oxygen, multicellularity would not be possible. We need oxygen to make lots of ATP. There's 36 to 38 molecules. And that's what's required for a complex multicellular organism to exist. Lots of ATP. If you're a single cell, we said you can get by on just glycolysis. You can do just fine with anaerobic respiration. But you'll do a lot better with aerobic respiration. And then you can associate together and form a multicellular organism. A real-life example that exists today is this organism here called Cyanophora paradoxa. It's a paradox because it's a eukaryotic protistine, so a eukaryotic organism, which has a prokaryote or prokaryote-like organism living inside of it, which happens to be dividing in this image. But this is an example of a eukaryote that has a prokaryote living inside of it. And this may represent what happened, countless millions of years ago and led to the evolution of mitochondria and chloroplasts. So any questions on the endosymbiotic theory? All right, so that'll do it. So it's about 3:30. Let's come back to lab at 3:40.